Today we're going to take what we've learned in previous modules and take a detailed look at the process of photosynthesis. So, looking at the overall equation of photosynthesis then, just a reminder then that it involves taking carbon dioxide and water, and that will be combined to form glucose, and oxygen gas will be released. And the molecule that's absolutely required for that is chlorophyll A, and this is an endergonic reaction. It requires an input of 686 kilocalories of solar energy. Now, photosynthesis is divided up into two sub-reactions, if you will. Each one is fairly detailed. One being the light reaction, sometimes called the light-dependent reaction, and the dark reaction, which also has other names such as the Calvin cycle, the three-carbon cycle, and some other names. All right, so we're going to be, when we look at photosynthesis takes place within chloroplasts. And so again, to orient ourselves, if we consider where a chloroplast is, and if you look at a tree, trees have leaves. Within those leaves, there are cells. Within those cells, there are chloroplasts. And it's within the chloroplast, then, that photosynthesis will occur. And when we look at the structure of a chloroplast, chloroplasts are composed of really two membrane layers, an outer membrane and an inner membrane. And this inner membrane is further subdivided or convolutes into what are known as thylakoid membranes. And so when you look at these diagrams from books or lecture outline, it'll look like there are actually three membranes, an outer membrane, an inner membrane, and thylakoid membranes. But there's actually a relationship between the thylakoid membranes and the inner membrane. And so I want to show you a more diagrammatic view of the chloroplast to show you these relationships. So again, there's an outer plasma membrane, there's an inner plasma membrane, and the inner plasma membrane actually then is continuous with the thylakoid membrane. So the thylakoid membrane isn't a separate membrane, it's actually connected to the inner membrane. And so we end up with these disc-like stacks of thylakoid membranes, and those are referred to as a granum. Singular would be granum, plural would be granus. So there would be lots of these within the chloroplast. And also notice then that this space between the thylakoid membranes, it's known as the thylakoid space, is continuous with this outer compartment. And then also this inner compartment contains a fluid known as the stroma. So when we talk about photosynthesis, we're going to be interested mostly in these thylakoid membranes because that's where the light reaction occurs. And then the dark reaction is going to be taking place in this fluid-filled inner compartment, the fluid known as the stroma. Now, light reaction. This is where we're going to begin. This is where photosynthesis begins. It occurs in the thylakoid membranes. This is where we have the conversion of solar energy to a usable form of chemical energy, ATP. And the products of the light reaction, and again we'll see this in a little while, are ATP, NADPH, and oxygen gas. And of those, oxygen gas is referred to as a waste product of photosynthesis. It will not play a role in other steps in photosynthesis, whereas ATP and NADPH are required for other steps of photosynthesis. So, looking at the light reaction, again, where it's going to be taking place is in these thylakoid membranes, these stacks of membranes within the chloroplast. And light energy, some light energy is going to be absorbed, some light energy will be reflected. And again, kind of looking at our stylized view here, the section of it, we're going to be interested in the thylakoid membranes, initially for the light reaction. And the pigments that are involved in the absorption of light are going to absorb mostly in the violet-blue range and also in the red-orange range, as you can see. And again, there's not much absorption in the green and yellow, and that's why when you look at a plant, it's green. That green light's being reflected. Other colors of light are what are being absorbed and driving photosynthesis. So, photosystems. We talked a little bit about this before, but I want to expand on our knowledge of photosystems. And again, what we're interested in here is the thylakoid membranes. Photosystems are an aggregate of somewhere between 200 and 400 molecules that include light-absorbing pigments known as accessory pigments. There are electron transport systems associated with the photosystems. ATP synthase is associated with these. Also chlorophyll A. And then there's something called the manganese center protein that we'll come back to in a few minutes. Now, getting back to these accessory pigments again, accessory pigments include chlorophyll B, carotenoids, and xanthophylls. And the accessory pigments are light-absorbing pigments. And what happens when they absorb a photon of light is that, again, that's something I've mentioned before, is that they will actually absorb a photon of light, and that will kick an electron out to an abnormal orbital. And as we've learned in talking about energy, the second law of energy tells us that energy will dissipate. So as that energy is lost, then the photon will return to its normal orbital, but it will pass that energy on then to an adjacent molecule in the photosystem. So when a molecule absorbs a photon of light, then, and the electron is excited, again, this energy can be passed from molecule to molecule to molecule. Now, again, getting back to what we're going to be talking about, we're going to be talking about the thylakoid membranes, and the first diagram I'm going to look at, we're going to be looking at the surface of a thylakoid membrane. So if we're looking at a top view of a thylakoid membrane, we will be seeing chlorophyll A's, accessory pigments, manganese center protein, ATP synthase, and electron transport systems. So here's our diagram. So again, the yellows are going to be accessory pigments. We have chlorophyll A's. We have electron transport systems. And we have ATP synthase scattered in here as well. And then also a manganese center protein. So again, initially here, what this is all about is absorbing light energy. And so these accessory pigments then will be absorbing photons of light. They get excited, pass that energy on to adjacent molecules, accessory pigments potentially, that also get excited. And so as photons strike these accessory pigments, they get excited, pass the energy around, and eventually we'll pass this energy on to chlorophyll A's. And so that's kind of what I'm trying to represent with these arrows is that this energy will eventually be focused on chlorophyll A's, exciting them. And so then that leads us kind of to the next step here. So what I'm going to do next is I want to look at just this part right here. We're going to look at this chlorophyll A and the electron transport system and the manganese center protein that's associated with it. Now, if accessory pigments have excited a chlorophyll A so that the chlorophyll A now has kicked an electron out to an abnormal orbital, and then it gets struck by a photon of light, that'll cause that chlorophyll A to actually lose its electron to the electron transport system. Now, what's important, however, is that that electron immediately needs to be replaced, and that's the role of this manganese center protein. The manganese center protein basically takes water molecules, strips hydrogens and electrons from one another, releasing oxygen gas. But the key here is the electrons that are made available. Those will replace the electrons lost by the chlorophyll A. If those electrons are not replaced by electrons from water, this electron just gets reabsorbed. And what's important is that loss of electron, that's the initiation, that's the beginning of photosynthesis. That's where photosynthesis begins. Now, we've talked about electron transport systems, and so the electron then gets passed through the electron transport system, and hydrogen protons will just kind of passively follow along. So, again, kind of replaying this again. An already excited chlorophyll A, so that's an important aspect. The electron has to be excited. Then if a photon strikes chlorophyll A, it loses an electron. That electron then will be replaced by electrons from water, and those electrons get passed through an electron transport system. And the hydrogen protons just follow along. The oxygen gas diffuses out of the plant and becomes part of our atmosphere. Okay, now next we're just going to look at these two systems here. So this chlorophyll A and the electron transport system, and this one with this electron transport system, and then look at that relationship. Now, I haven't mentioned this, but there are two types of chlorophyll A's. There are what we call P680s and P700s. And they absorb light at different wavelengths. This absorbs light of 680 nanometers, and this is light of 700 nanometers. And we refer to this chlorophyll A and its electron transport system as a photosystem 2, the P700 and its electron transport system as a photosystem 1, or part of photosystem 1. And I don't know why they're really ordered that way, because it really begins with photosystem 1 and begins with photosystem 2 and continues with photosystem 1. So again, we're going to basically be looking at the same kind of thing. What happens is that an excited chlorophyll A, in this case P680, gets struck by a photon, it loses the electron. And so that electron then gets passed into the electron transport system, but of course that has to be replaced by an electron from the breakup of water. And then that electron will be passed through an electron transport system, and it gets to the end of that chain. And it basically has nowhere to go until this chlorophyll A loses an electron. Now once that chlorophyll A loses its electron, this electron can replace the one lost by the chlorophyll A. So it'll take the place of the lost electron, and now this can be excited and more electrons can be discharged from that. And so the electron then gets passed through the second electron transport system, where finally a terminal electron acceptor, NADP, will pick up that electron and it's reduced to NADPH. So I'm just showing this as one electrons, but really electrons are passed in pairs for the most part. So that's another thing to keep in mind. So just one more look at this then. Chlorophyll A in photosystem 2, already excited, accepts a photon, loses its electron to the electron transport system. That has to be replaced by electrons from the manganese center protein, stripping electrons from water. That electron is passed along, will replace the electron lost by the P700 of photosystem 1, and then those electrons get passed through that electron transport system and picked up by NADP, which is reduced to NADPH. So when we look at this flow of electrons in, the source of electrons is water molecules. The terminal electron acceptor is NADP, which is getting the electrons out. And that will then give us flow of electrons. Okay, now what I want to do is really look at this system again, these two electron transport systems, but now from a side view. Let's look at the thylakoid membranes from the edge view rather than from a surface view. And so again, we're going to be looking right in the side of the thylakoid membrane now. And so again, we've got our photosystem 2 or photosystem 1, but we've added also an ATP synthase here. And also just to orient you where we are, this would be the stroma. This is the thylakoid space. So chlorophyll A is going to get excited, struck by a photon of light. It's going to lose the electron. It's going to be replaced by an electron from the breakup of water. That gets passed through this first series of electron transport systems. And again, this is the photosystem 2. And what happens here is that's where the hydrogens are pumped. So there are hydrogen pumps in this electron transport system. And then, again, the electrons continue on through the electron transport system of photosystem 1 and are eventually picked up by NADPH. So this second series is really just to position the electrons for pickup by the terminal electron acceptor. So again, source of electrons, water, terminal electron acceptor that gives us flow of electrons, which pumps hydrogens. Why do we want to pump hydrogens? So that they can pass through the ATP synthase. And of course, when hydrogens go through 2 at a time, what gets produced? ATP is produced by the ATP synthase. So again, what we have here is an oxidative phosphorylation. So the overall effect then is to take solar energy and generate chemical energy in the form of ATP. So again, one more look. So electron lost, replaced by water. As that electron flows through the photosystem 2 electron transport system, that causes pumping of hydrogens. Electrons continue through photosystem 1 so that they can be picked up by the terminal electron acceptor NADPH. That causes pumping of hydrogens. Hydrogens concentrate, follow their concentration gradient through the ATP synthase. ATPs are made. And again, just a diagram, another diagram to show the same kind of thing. I don't think this works actually as well as what we just talked about, but again, this is going to be in your lecture outline if you want to refer to this one. Okay, now, the other side of photosynthesis then will be the dark reaction, also known as the Calvin cycle. So here, carbon dioxide and water combine to form glucose. This requires energy from the light reaction, or requires certain materials from the light reaction, including ATP, so energy in the form of chemical energy, and NADPH, so electrons and hydrogens from NADPHs are also required for the dark reaction or Calvin cycle. Products of the Calvin cycle include glucose, ADPs and phosphates, and NADP. So the ADPs and phosphates and NADP can then also be reused then by the light reaction. Now the dark reaction is going to be taking place in the stroma of the chloroplast, so not within the thylakoid membranes or space, but outside of those. So it's going to be in this area. And essentially this is a bag of enzymes. And so the dark reaction, or the Calvin cycle, then is an enzymatic cycle. And there are essentially three parts to this cycle. One is the fixation of carbon dioxide, followed by the actual glucose production, and then regeneration of some important molecules known as RUBPs, which stands for ribulose bisphosphates. Now keep in mind that when we talk about a cycle, then we need to start and end with the same materials. And what we're going to start and end with then are ribulose bisphosphates. So let's start with the fixation of carbon dioxide. Fixing carbon dioxide, what that means is that when you fix a molecule, you're going to take a molecule and attach it to an already existing organic molecule. So keep in mind that carbon dioxide is an inorganic molecule. And the purpose of photosynthesis is to generate organic molecules from inorganic sources, carbon dioxide and water, into glucose. So what's going to happen here is that carbon dioxides are going to be added to molecules called ribulose bisphosphates. So we're going to start with 6 ribulose bisphosphates. And 1 carbon from carbon dioxide will be attached to each of those ribulose bisphosphates to generate 6 carbon intermediate molecules. So the ribulose bisphosphates are composed of 5 carbons each. We're going to add a carbon from carbon dioxide to each of those. So we'll end up with 6 carbon molecules. And the enzyme that accomplishes this is something called Rubisco. It stands for ribulose bisphosphate carboxylase. And it's a very interesting enzyme, of course, because it fixes carbon dioxide from the atmosphere into an organic molecule. And it's also fairly inefficient, to be honest with you. Most enzymes work at about 20 to 30,000 cycles per second, and it only carries about 2 to 3 reactions per second or cycles per second. Okay, so let's go ahead and look at this fixation then in a little more detail. So we're going to start then with 6 ribulose bisphosphates. Ribulose bisphosphates are a type of sugar, 5 carbon sugars. So we're going to start with those. And then to make 1 glucose, you're going to need 6 carbons, so that's going to require 6 carbon dioxides. So what Rubisco will do then is take a carbon from carbon dioxide and attach that to a ribulose bisphosphate. And then it will do that for a second one. So it's going to take 6 ribulose bisphosphates to generate a single glucose. And then a third. And then a fourth. And then a fifth. And then a sixth. So at the end of this part of the cycle then, you start with 6 5-carbon ribulose bisphosphates. You end up with 6 carbon intermediates, and these are very unstable molecules. Now, the next phase then is to actually produce glucose, so glucose production. So what that involves then is taking our 6 carbon molecules, splitting those in half through a series of reactions into 3 carbon molecules. So if you're going to go from 6 carbon molecules to 12 3 carbon molecules, called G3Ps, or glyceraldehyde 3-phosphates. And that will also require 12 NADPHs and also 12 ATPs, both coming from the light reaction. Now, of those 12 3-carbon molecules, of those 12 G3Ps, 2 of those will be diverted then to producing glucose. So if I take 2 3-carbon molecules, those 6 carbons can be used to make glucose. And then the other 10 will be used to regenerate ribulose bisphosphates so that we end up with what we started with. So let's look at this in a little more detail then. So again, we're going to start with our 6 carbon intermediates. They're going to be split in half to produce 12 3-carbon molecules called glyceraldehyde 3-phosphates. That's going to require ATP, or excuse me, NADPH and ATP. So again, those split into those. And then 2 of those will be diverted into glucose production. So molecule glucose will be produced. So 6 carbons go out. And then the remaining 30 carbons will be used to regenerate ribulose bisphosphates to get back where we started. So we're going to start with 36 carbons. they're going to be still 36 carbons, just in a different form. Instead of 6 carbon molecules, they're going to be 3 carbon molecules. 6 of those will be diverted off into a pathway to make glucose, and the remaining 30 carbons then will be used to regenerate RUBPs. So last step then is regenerate the ribulose bisphosphates. So in the regeneration of ribulose bisphosphates, the 10 glyceraldehyde 3-phosphates, through a series of enzymatic reactions, will be reassembled to form 6 ribulose bisphosphates, each for 5 carbon sugars. So we're going to start with 30 carbons, we're going to end up with 30 carbons, but they're just going to be in a different format, and that also requires 6 ATPs to carry out that series of steps. So again, we're going to take our 10 glyceraldehyde 3-phosphates, and they're just basically going to be reassembled then into ribulose bisphosphates. So we're going to take 10 3-carbon sugars, and they're going to be assembled into 6 5-carbon sugars, which are the ribulose bisphosphates. And that gets us back to where we started in the cycle. And of course, that's also going to require 6 ATPs. So as we look at the cycle then, we start with 6 ribulose bisphosphates. We go through a series of changes. We're going to bring in 6 carbon dioxides. So we start off with 30 carbons. We're going to bring in 6 carbons to make 36 carbons in the form of 6 6-carbon molecules. Those are going to be split up into 12 3-carbon molecules. 2 of those are going to be diverted off to make glucose. The other 10 are going to be used to regenerate the ribulose bisphosphates. So a typical cycle. We're going to start and end with something, bring materials in on one side, take materials out on the other side, and then just keep it going. And again, understand it's a number of different reactions, not just a few reactions. A lot of enzymes involved here. So again, another look at the same idea. We're going to start with 30 carbons in the form of ribulose bisphosphates. We're going to bring in 6 carbons in the form of carbon dioxide, so that gives us 36 carbons. Those are going to be transformed into glyceraldehyde 3-phosphates. And of those 36 carbons, 6 are diverted off to glucose production, and the other 30 are reformed into ribulose bisphosphate. Along the way, you're going to need 12 NADPHs. You're going to need 12 ATPs here and 6 more ATPs here. So to carry this whole cycle out, one turn of the cycle to generate one glucose molecule then is going to require 12 NADPHs and 18 ATPs. Now, also, as we look at the relationship between the light reaction and the dark reaction then, the light reaction generates ATPs and NADPHs that are required for the Calvin cycle, and then the Calvin cycle will generate NADPs and ADPs and phosphates, which can be put back together to drive this more. And then the other thing, of course, is that CO2 has to be coming in from the outside for this to work. So again, the light-dependent reaction absolutely requires light. You have to have light for the light-dependent reaction. So the so-called dark reaction or Calvin cycle can occur in the light, but it doesn't really require light. It just requires ATP. So these enzymes are ATP dependent, not light dependent. And as it turns out then, the light reaction is what's going to convert that solar energy to chemical energy of ATP. Okay. Now, one other thing I want to talk about is where the CO2 comes from. So water, we need water and carbon dioxide for photosynthesis. Water is going to come from the roots. That's going to be brought up through the roots and delivered throughout the plant. Carbon dioxide has to come from the atmosphere, has to come from the air. And so as it turns out, when we look at the bottom surface of leaves, there are little pores in the bottom surface of the leaf. And this is how carbon dioxide gets into the interior of the leaf and eventually into the cells. And leaves are covered in a waxy cuticle, which prevents water loss, but also prevents gases from penetrating as well. So these pores are important. So as we look at these then, there are these cells known as guard cells, which create an opening in the leaf known as stomata. Singular would be stoma, plural is stomata. And those stomata can either be open or closed. So again, this would be a stoma that's open. So the guard cells are allowing the stoma to be open. Now, it's important for the stoma to be open because that allows CO2 in, but that also allows water to escape. So, as it turns out, these guard cells are kind of kidney bean shaped. And the more hydrated they are, the more they bend. So, when a plant is fully hydrated, the water moves into the guard cells, they bend more, and that opens up the stomata. And they actually have mechanisms to move sodium into the cells, draw water into the cells. So usually the stomata open up in the morning. Then as the day progresses, as the guard cells dehydrate, and as the plant dehydrates then, the guard cells sag together. And that closes the stomata. And so this is what it looks like when it's closed. Now, closing the stomata then prevents water loss, prevents the plant from getting too dehydrated, but by the same token then, it loses out on the ability to absorb carbon dioxide. Now, so again, fully hydrated, stomata open, dehydrated, guard cells, stomata closed. Now, there are some plants then that have a modification to try to get carbon dioxide into plants very rapidly, and this occurs in plants that are adapted for drier habitats. And this is what's known as the C4 pathway. So in the C4 pathway, what happens is that there's a 3-carbon molecule that will bind, that will have an enzyme that will bind incoming carbon dioxide to that. So these are carbon dioxides, and they will bind to a 3-carbon molecule to form a 4-carbon molecule, hence the 4-carbon pathway. Then that will follow its concentration gradient into an adjacent cell, and there that cell will give up the carbon dioxide to the Calvin cycle. And then this 3-carbon molecule will follow its concentration gradient back to the original cell where it can pick up more carbon dioxide. So again, this is known as the 4-carbon pathway because a 4-carbon molecule is formed here. And then that will give up a carbon dioxide in the adjacent cell and then follow its concentration gradient back. So the point of this then is that as carbon dioxide comes in, it's bound up to a molecule. That molecule then moves out of that cell into another cell. It gives up the carbon dioxide there. So that keeps carbon dioxide levels within the cells low. So carbon dioxide diffuses rapidly into the cells. That keeps carbon dioxide levels in the airspace between cells low, so that draws carbon dioxide in from the outside world into the leaf as well. So what this C4 pathway then does is allow for rapid concentration and assimilation of carbon dioxide so that in the morning hours, carbon dioxide can be brought into the plant quickly. Then when the plant dehydrates, the stomata close off. And then there are some other plants called chem plants, which also have a C4 pathway, but they have an additional adaptation in which they actually open the stomata at night and close them in the day. And an example of those kind of plants would be cacti that we see out in the desert. So we've learned a lot. That does it for photosynthesis. I'll see you next time.